So since we're convinced that natural selection is in action and that changes in allele frequencies are occurring, the next step would be trying to understand exactly how natural selection affects traits. And it depends on how the trait itself is determined. Is it a single gene trait or is it a polygenic trait? And we're going to take a look at both of those. The single gene traits are a little bit easier to understand, so we're going to start there. Remember, single gene traits are controlled by two alleles. You get one from mom, one from dad. And because you can only have two possible alleles, they can either be two of the same or two different. And the bottom line is, in most cases, we're only going to get two different phenotypes because you're either going to have the dominant phenotype or you'll have the recessive phenotype. Sometimes you can have three phenotypes, though. If we're talking about an example of incomplete dominance or codominance, we can sometimes see a third phenotype as well. We're going to focus on the three different types of phenotype because that's as complicated as it gets, but it's still pretty easy to understand, I hope. And if you understand what happens with three, you can certainly understand what's going to happen with two different phenotypes. So the example we're going to use is a population of birds with varying beak sizes. So the thing that's varying within the population that we're concerned about is beak size. We're going to use the red color to indicate birds that are homozygous for a large beak. And if they are homozygous, meaning they have two alleles for a large beak, they're going to have large beaks. We're going to use yellow to indicate birds that are homozygous for a small beak, meaning they have two alleles, both for small beak, and therefore they need to have small beaks because that's the only alleles that they have. And we're going to use blue to indicate heterozygous birds that have an allele for a large beak and an allele for a small beak. And we're going to assume it's incompletely dominant, and that gives us a medium beak. We're somewhere in between the two others. So if we take a look at our original population, and this will be our original population right here, and we can see that the number of birds with different beak sizes is approximately even. This would indicate that originally in this particular population there was no favored beak size, that all three beak sizes would have had equal success surviving and reproducing. But let's say that for some reason in the environment a change occurred, and as a result of that change the population distribution changes. Let's say that all of a sudden the population has an abundance of large seeds, and because of the large seeds only the birds with the large beaks are able to eat well and reproduce. The birds with the medium-sized beaks and the smaller-sized beaks can't eat the seeds, they don't get the nourishment, and they die before they're able to reproduce and pass on those genes. So the only thing that's going to be able to pass on their genes over generations is those that have two genes for large beak. Over time, we're going to see more and more birds with large beaks, and we're also going to see more and more alleles for large beaks. So essentially what's happening is that one of the phenotypes is favored, and the other two are not. And that's typically what would happen in a single gene trait. One of the phenotypes is going to be favored, the other one or the other two in this particular case are going to fade away. It's really, really tough to get them to fade away completely so that the other allele is completely out of the population, especially if it's recessive. So typically, I'm not really comfortable saying that one allele is going to completely disappear. It's certainly going to be reduced and have minimal impact, but to say it'll disappear would be a bit of a stretch. But, so bottom line, this is pretty easy. What's going to happen is one of the phenotypes is favored, becomes more common, the others are significantly reduced. The polygenic traits are a little bit more tricky. When you look at polygenic traits result in multiple phenotypes. And we talked about examples in class, things like skin color in humans, height in humans. Those are things that are determined by multiple genes. And by the time you figure out all the effect of all the multiple genes, you have this wide range of phenotypes. So this particular example is using coat color in mice. And as you can see, we range from really, really light mice to really, really dark mice. And the way the distribution works, it works out in a bell-shaped curve, normally. What happens is that we see very few mice that are really, really light, and very few mice that are really, really dark. Most of the mice fall somewhere in between, and then there's this steady falling off on either side from the middle. what can happen is that we can have a couple of different types, actually 3 different types of selection against polygenic traits. We can have stabilizing selection, directional selection, and we can have disruptive selection. And the difference is exactly which part of the bell curve is being favored. So let's take a look first at stabilizing selection. In stabilizing selection, what's happening is that the extremes are being selected against. And as a result, we see fewer and fewer of the extremes, and we see more and more of the middle. And so our bell-shaped curve becomes a very, very narrow bell-shaped curve, not a very broad one. So as a result, we're stabilizing things, and things are becoming more, average is becoming more popular. The extremes are starting to fade away. So essentially, we're stabilizing the population to the average. Directional selection is a little bit different because instead of selecting against both extremes, we only select against one extreme. In this particular example, we're selecting against the lighter colored mice. So over time, what happens is we begin to see more and more darker colored mice. So our bell-shaped curve is now moving to the other direction. That's why it's called directional selection. This could also work the other way. We could have had something against the dark colored mice instead of the light colored mice, and then our bell-shaped curve would have gone the other way to favor the light colored mice. So it can happen in either direction, but the key is this: it's only going to happen in one direction. That's why it's called directional selection. If it happens in both directions, well, sorry about that, I meant to erase it. If it happens in both directions, that's called stabilizing selection. So when it's only happening on one side, that is directional selection. The last example of a type of selection we can have is called disruptive selection, and that's this one right here. And as you can see in disruptive selection, this is the only option that's left. And this is when the average, or the middle, is actually selected against. As a result, we begin to see more of the extremes. In this case, we see more lighter colored mice and more darker colored mice, but not so many of the ones in the middle. That's called disruptive selection. So when it comes to polygenic traits, things are a little bit more complicated because we have a whole lot more phenotypes. There's a whole lot more variety. There's more than just 3 possible phenotypes. But there's still only 3 possibilities of things that can happen. We can have stabilizing selection, directional selection, and disruptive selection.